Hi everyone, this is part one of videos devoted to epithelial tissue. In this video, we will discuss what are the types of epithelial tissue and what are the features that differ it from other tissues in human body. So let's go. First thing first, we need to understand that epithelia can be of two types, either lining or secretory. In this video, we'll be focusing on lining epithelia. So it can line either our external skin epidermis, it can line internal cavities like mesothelium, like endothelium in blood vessels, or it can line our tubes and cavities inside our body, either in gastrointestinal tract, respiratory, or genitourinary tract. But despite what type of epithelial we have, lining or secretory, it will be having some common features. First of all, this is cellularity. So there is no extracellular matrix, and because of this, we have specific junctions in between cells. We'll talk about them later on. Secondly, if this is simple epithelium, all of the cells will be joining to basal lamina. So epithelia cannot exist without basal lamina. If it's stratified epithelium, then only basal lowest level will be attached to basal lamina, and therefore all of the other layers will be coming up above, above, and above. So, presence of basal lamina, cellularity, no extracellular matrix, specific type of junctions in between cell, and polarity. So, what does polarity in epithelium stands for? On the screen, you see right now simple columnar epithelium, so tall cells. You see that their lower bottom part is attached to basal lamina. And this lower part of the cell we call basal pole because it is attached to basal lamina. And the opposite part, the apical part, is called the apical pole. Why do we need that? Because if we say the basal pole of the epithelia, we mean that there is nucleus and some of the compartments and organelles. If we say apical epithelia, this is part of the cells that we basically may have microvilli, stereocilia, or cilia, and therefore we will have specific type of epithelia. For example, ciliated epithelia, if on the apical pole we have the cilia. And the second important part is that through the basal pole of epithelial cells, we have the nutrition of epithelia. Epithelia does not possess any blood vessels. So how does it survive? It survives by diffusion of nutrients from loose connective tissue that is basically located below the basal membrane. That means below the basal pole of epithelia. And by diffusion, we have all of the nutrients coming up to epithelia despite what type we have, secretory, lining, simple, or stratified. So polarity does mean a lot functionally and clinically. As we mentioned, through the basal pole, we have the nutrients coming up to epithelia from loose connective tissue because epithelia itself is avascular. So what about the apical pole? If we have the microvilli on the apical pole, as you see on the screen, this basically means that this cell is able to absorb something. And such type of epithelia is predominantly present in small intestine. This suggests the projections of plasma lemma with no specific formulas of microtubules or microfilaments in it. If the cell has stereocilia on its apical pole, we basically mean that this is a sensory cell because such cell we can find, for example, in organ of corti in our internal ear. So it will have some specific frequency of movement for this stereocilia. And lastly, on the apical pole, our epithelia can have just cilia. So these are not stereocilia. Cilia are, for example, found in upper respiratory tract, and they also have the movement, but this will be the movement against the inhaled air. So I've started with morphological features of epithelium to show you and persuade you that they do have importance in clinical concern because the fact that they will have specific type of organelles on their apical surface and whether this epithelia will be simple or stratified, it will carry out some specific and important functions for our body. So it can be secretion of internal or external type. This can be absorption like columnar epithelium with microvilli in small intestine. This can be transport in urinary system and respiratory tract. It can be protection like stratified squamous keratinized epithelia of our epidermis, and it can be receptor function like that cell with stereocilia in organ of corti in our internal ear. So what about the junctions? As lining epithelia basically creates borders between cavities and environments in our body, it will have some specific junctions. They are of isolating type in between cells. Plus, we do not have any extracellular matrix, so we don't have the communication with it. So the types of junctions that predominate is desmosome. And to attach epithelia to basal lamina, because basal lamina is the fundament for our cells, we have hemidesmosomes. So this means that we split those two protein plaques in a half because it basically comes from each of the cell, each half of the desmosome. So we have only one half from the basal pole of our epithelial cell, and it attaches through the core proteins to our lamellin proteins in the basal lamina. Classification of lining epithelium is based on the fact how many layers of cells we have. If it's only one layer of cell, that means a single layer of epitheliocytes, all of them are attached to basal lamina, we call such type simple epithelia. And depending on the shape of the cells, simple epithelia can be either squamous, the cells are flattened, either cuboidal, the height and the length of the cell is equal, either columnar. Plus, we have columnar pseudostratified epithelium. We'll talk about it later on. Contrary to simple epithelium, if we have two layers and more of epithelial cells, we call such type of epithelia stratified. Depending on its functional properties, stratified epithelia can be keratinized, non-keratinized, and transitional. Depending on the superficial layer of cells, despite the fact these are 5 layers of cells, 2 layers of cells, 10 layers of cells, it can be also squamous, cuboidal, columnar, only depending on the superficial layer of cells. Let's take a look at each of these type of epithelia. Starting with simple epithelia and simple squamous epithelia, the cells are flatter, there is only one layer of cells, and they have really irregular borders. There are two good examples of a simple squamous epithelia. First is endothelium. This is the specific name for squamous cells lining our blood vessels. And second type is mesothelium. These are sometimes secretory cells that are lining our internal cavities, and for example, they're lining peritoneum or pericardial cavity. Simple cuboidal epithelium has equal sizes of its wall, and the nucleus therefore is located in the center of the cell, which is very easy to identify under the microscope. And it is really commonly found in our body, specifically lining the excretory ducts of different glands. Simple columnar epithelium consists of a single layer of tall elongated cells, where nucleus is located classically on the basal pole of epitheliocytes and on the apical surface. These epithelia, depending on their localization, may possess microvilli, stereocilia, or cilia. Such type of epithelium can be found in gastrointestinal tract, like in stomach and small intestine and large intestine, in fallopian tubes, and in airways as well. Pseudostratified epithelium is a single layer of cells. All of them are attached to basal lamina, but because they're of different shape and sizes, their nuclei are located on different levels, which gives us the appearance of multi-layered. But morphologically, it is one layer of cells. Some cells that I want to identify here are the basal cells. They're responsible for regeneration, goblet cells that are responsible for mucus production, and just ciliated cells that are responsible for clearance of inhaled air. Such epithelium is also called respiratory epithelium because it is mostly found in upper airways. Now let's move to three types of stratified epithelium. First representative is stratified squamous keratinized epithelium, and it is located in the epidermis of our skin. That means the upper layer of our skin. Morphologically, it consists of 5 functional layers: stratum basale, stratum spinosum, stratum granulosum, stratum lucidum, if it is thick skin, and stratum corneum. What happens here is the cells, starting from the basal layer, undergo maturation and keratinization as they move purposefully to the upper layer. Therefore, if we talk about thick skin, stratum lucidum and stratum corneum are the dead cells. They do not possess nucleus, but this layer creates a really good border from the external environment. Stratified squamous non-keratinized epithelium can be found in oral cavity, cornea, and vagina, the biopsy from which you see right now on the screen. It has many layers morphologically, but only 3 layers functionally. That means basal layer, intermediate layer, and superficial layer, and it does not undergo keratinization. That is why, in case you are confused whether this is keratinized or non-keratinized epithelium, you can always zoom in the most superficial layer that is facing the cavity or external surface, and you will see that these cells possess nuclei, which means they are not dead and they do not undergo keratinization. Can they undergo keratinization? Yes, but this will be a pathological process. So normally, it does not undergo keratinization. Last but not least, transitional epithelium. It is a multi-layer type of epithelia where a superficial layer of cells has some specific properties. They are called umbrella-like cells or dome-shaped cells. Right now on the screen, you see biopsy made up from empty urinary bladder, and the cells are relaxed and big and extended. Whenever it becomes full with urine, they become flattened and vice versa. So this is the specific feature of this type of epithelium that is located exclusively in the urinary tract. Thank you for watching. I hope this was useful and see you in next video.